Hello student, today we are going to start on the topic of cleavage in human embryo. Let's first define the cleavage. Cleavage is a succession of rapid mitotic division just after fertilization, which converts fertilized ovum of exceptionally large cytoplasm nucleoplasm ratio into a multicellular mass of blastomeres called morula. The word exceptionally large cytoplasm nucleoplasm ratio is very important. It means actually the size of ovum is far larger than a normal body cell, and that ratio has to be maintained. And actually, the cleavage is process. The one aspect of that is to maintain that ratio. So usually it begins 30 hours after the fertilization. You know that fertilization takes place in the ampullary part of the fallopian tube, and after the ovum is released, it has been seen that earliest the start of cleavage is about 30 hours after fertilization. So normally it occurs as zygote pass through the uterine tube after the fertilization, of course. During cleavage, the zygote lies within the zona pellucida. There is little overall cytoplasmic growth, but dramatical large growth of DNA. So actually, the importance of zona pellucida is that as long as it is there, it doesn't allow the developing zygote to stick to the wall of uterine cavity. Otherwise, it can lead to a tubal pregnancy. And another aspect is that there is little overall cytoplasmic growth, means there is no growth in cytoplasm, means the size, the volume of the zygote will not increase, but the volume of the DNA will increase. Means the nuclear contents will increase in their number. The overall volume of the nuclear content will increase at the expense of the cytoplasmic contents. So how it happens, we'll learn it later on. DNA in successive blastomere is not formed by transcription of mother blastomere. Actually, this is an important aspect that during the cleavage, the original DNA of the mother zygote, mother cell in the zygote, that remains in the form of heterochromatin and uncoiled material of inactive DNA and doesn't participate in the cellular division. So from where these new DNA are coming up, that we'll also see actually they come from the mRNA present within the cytoplasm itself. Cleavage. There are different kind of cleavage depending upon the different kinds of cell. The cell in different organism vary depending upon amount of yolk sac present in it. The organism, in particularly the egg-laying organism, where whole embryo development take outside the body of organism, they are laden with, those eggs are laden with large amount of yolk. But humans, because the most of embryo development take place within the body of mother, so they need a small amount of yolk sac, and such cell they are isolecithal, wherein the yolk sac is also equally distributed throughout the cell. And in such cells, the pattern of cleavage has three characteristics, that they are, the cleavage is holoblastic, it is rotational, and it is indeterminate. Holoblastic cleavage means that the cells after the division of the zygote, we call those cells as the blastomeres, they are completely separated from each other. In certain organisms, they are not completely separated but partially attached to each other. Such a division is termed as meroblastic. But in human beings, the cells are completely separated from each other, so it's a holoblastic cleavage. Another characteristic is that the cleavage is rotational. Cleavage can be transverse, it can be longitudinal. In humans, it is rotational, means alternatively, there's one longitudinal cleavage, then there is a transverse cleavage. And another characteristic, still another characteristic, is indeterminate. The determinate means that when the cells, the blastomeres formed after the zygote, when those cells, they are special, means each cell is determined to form a particular part of embryo, then the division is determinate. But human cells, particularly during the cleavage, they are indeterminate in nature, means the cells, they are, all the cells, they are capable of forming the complete embryo. They are not determined, means they are not forming a particular part of the embryo. So cleavage type in humans is holoblastic, that is complete. It is rotational, means cells alternately divide as in transverse and longitudinal direction, and it is indeterminate, means cells are capable of forming complete embryo. Cleavage starts after fertilization. Let's know a bit about fertilization. From our previous study of oogenesis, we know that human ovum is isolecithal, as we just discussed. It is released from the ovary at the stage of primary oocyte. It is captured by fimbria of fallopian tube, as evident from the diagram by the blue arrow, and it is carried to the ampullary part of the fallopian tube. Ampullary part is the most dilated part, as we can see, as far as dimension is concerned, of the ovarian tube. And it is here actually the sperm meet with the ovum to cause fertilization. Now this fertilization of ovum, it is dependent upon many factors. We know that the ovum, it is surrounded by zona pellucida and few cells of the corona radiata, which are protective in nature. As long as zona pellucida is there, it will not allow the fertilization to take place, as we discussed earlier. So as many sperms are present around it in the ampullary part of fallopian tube, they try to pierce through the zona pellucida. Finally, one of them is capable of doing that. And once it enters inside, pierces the zona pellucida, it also passes through the vitelline membrane of the ooplasm. Once a sperm is poured the contents in the ooplasm, the zona pellucida undergo a reaction which is called as Spona reaction, and by virtue of that, entry of any other sperm is disallowed in the ooplasm. So finally, the sperm pour the contents which are present in the head of the sperm, that is the nucleus part, into the ovum and in the form of pronucleus. So it enters inside ovum, and this is a right time for the zygote formation. In this particular slide, we can see that the two nuclei are present side by side within one cell. These are male and female pronucleus. One, they are termed as pronucleus, means they don't have very well developed nuclear wall. In fact, they are already in the process of forming the spindle. And male and the female pronucleus, they are lying side by side. So some workers, they like to say it 1.5 cell stage, because cell is not completely fused. Of course, there is a single cytoplasm that is of the mother origin, that is of the egg origin, but the two nuclei, they are still separated from each other and are not fused. So certain workers, they like to call it 1.5 cell stage. So this is a zygote with two pronuclei present within it, ready to divide. First cleavage division is the resultant of the zygote. The cells, they divide along the vegetal animal pole and divide into two. And these two cells, they are termed as blastomeres. And these blastomeres, each of them is a complete cell in itself, that each of them is capable of forming a complete embryo. It's seen about 30%, roughly about 30%. Cases of monozygotic twins are actually the separation of these two cells at the first cleavage division, that these cells, they completely divide and start developing separately. Each of them capable of forming a complete embryo. So ultimately, two embryo they start developing inside the female uterus. So first cleavage division brings about two blastomeres, uh, and again, volume of the ovum as such doesn't increase. The cytoplasm of the original ovum get equally roughly get divided into two cells. But the nuclear content, it has doubled. There are two nuclei now. Second cleavage division is along the axis which is perpendicular to the axis of the first cleavage division. So this is in true spirit of the rotational cleavage division as in the human beings. Four blastomeres are formed. Still, they are undifferentiated, means all are identical. Again, we see there are four nuclei now, means the nuclear contents have increased, but there is same amount of cytoplasm. Within it, within this slide which we can see the zona pellucida intact all around, and many sperms, uh, uh, dead or alive, they are seen around, entangled within the corona radiata of the cell. Third cleavage division. Uh, 8 blastomeres are finally formed, and at this particular stage, it is found that usually there is one central blastomere which is surrounded by the 7 peripheral blastomeres. And though most of the time, if one blastomere is removed, the embryo is completely formed, but occasionally it may happen that if one of the cell is removed, the embryo is not formed, and the further development is only of a trophoblastic shell, means no embryo inside, only the coverings of the embryo, they are formed. It happens sometime. So it suggests that some kind of differentiation has started within, that one of the cell maybe uh, has taken up the potential of forming the embryo, the other one, the coverings of the embryo. All the cells, they are still independent of each other. They are lying close to each other, but they are not sticking to each other. That can be understood because these cells, they show relative movement to each other. There is a large amount of movement there, and it also shows a lot of activity is going inside these blastomeres. Further, the cells undergo divisions and a ball of cell is formed. Uh, but an important thing happens during the fourth cleavage stage. That is when the cells, they duplicate from 8 to 16. During this time, the cells are not showing relative movement. They tend to adhere with each other. Such a phenomena is called as compaction. Now the cells, they have oriented themselves. They do not show and they cannot show a relative movement from each other. Uh, and this is an important point which starts that there is an onset of differentiation. Now the cells, they are differentiating and they are destined to form a particular structure. In one of the developing cell in the morula, the 12 blastomere stage, one cell is found to be central, which is going to be the mutual embryonic cell, and 11 of the cells, they are peripheral in nature. It suggests that they will form the trophoblastic layer of cell. It is initiation of two diverging lines, the embryonic or the formative cells, and the other one is the trophoblastic cell. Further, this ball of cells, the morula, it will form a blastula with a cavity finally, which is called as blastocyst. Uh, in these particular diagram, as we see, the morula is a solid ball, generally up to 32 cell stage, uh, developing a cavity slowly, which is formed by absorption of the nourishing fluid from the stratum compactum of the uterine endometrium, because by fifth day we know that this morulite lose pellucida and start invading the stratum compactum. So before becoming blastocyst, means from morula to blastocyst, it undergoes certain physiological changes within itself, leading to a physiological stage, which is called as MBT, the mid-blastula transition. So what is this mid-blastula transition? Actually, this stage marked the end of the cleavage. Let's understand it. Mid-blastula transition is a stage which marks the initiation of mRNA formation by blastomeres. Now, what is this? In initial slide, we had discussed that in earlier stages of the division during the cleavage, the uh, nuclear contents which are formed in the successive blastomeres, they are not of the mother origin, because the mother DNA, it is in the form of a heterochromatin which is inactive in state. Then how the new DNA is being formed? It is being formed by the mRNA which are already present in the cytoplasm of the ovum. It means the precursor material for formation of these mRNA is also present within the cytoplasm. So cleavage goes as long as that mRNA which is present in the cytoplasm is capable of forming the new nuclear material. And it keeps on doing it till an appropriate ratio of the normal cytoplasmic nucleoplasmic contents is reached, that is about 6%. So is it that cleavage is a process to bring about the very large ovum into the normal body cell, into the size of the normal body cell? So the mid-blastula transition, when this ratio is reached, when the original DNA is no more heterochromatin but become active and actively start dividing, transcripting to form new DNA, that marks the end of the cleavage and start of the blastula or the blastocyst. We can understand few characters of the cleavage from this understanding, that cleavage in fact is a slow process. It takes about 4 to 5 days to form the morula. And another thing is the formative cells grow slower than the trophoblastic cells. It is evident from an isolated specimen, which was 107 cell specimen of blastomeres, in which about 60 cells were forming peripheral layer, the trophoblastic layer, and in fact 69 cells, and 30 another cells were small trophoblastic cells which were due to form the trophoblastic layer. And it was seen that only 8 cells, they were formative cells, means only out of 107 cells, 8 cells will form embryo, and rest of the 99, they were responsible for the formation of the layers of the embryo. And another important thing which we can understand from this discussion is about IVF, the in vitro fertilization. We know this is a procedure which is adopted in the females who do not have the patent female reproductive tract. So where it is, what is done, an ovum at the primary oocyte stage through the suitable methods is procured from the ovary, and that is made to fertilize it with the sperm in vitro, that is experimental condition, in the test tube. And then that fertilized ovum is implanted into the uterus, which usually get implanted in the posterior upper part of the uterine cavity. So when it is implanted, generally the zygote is at the stage of second cleavage or sometimes third cleavage. So it is during this stage the zygote is implanted. So IVF has ability to provide progeny to the individual, to the females who otherwise do not have patent reproductive tract. A more evolved form of it, the gift which is another advanced IVF technique by which there are about chances of 40% or more than 40% for the pregnancy. The classical IVF only provides less than 20% chances. Now, we can make a summary of the cleavage process with the help of this diagram. What we see in this diagram originally, the ovum at the primary oocyte stage is being obtained from the ovary. So as it ruptures, the surface of the ovary rupture, it is captured by the fimbria of the fallopian tube, start moving towards the ampullary part, where it receives many sperms on the day one itself. By the day two, the fertilization take place and the cleavage initiates. And by the day four, a ball of morula is formed which reaches the uterine cavity. And by fifth day, it starts penetrating into the stratum compactum of the uterine endometrium. So there's a large increase in the number of the trophoblastic cells almost explosively, whereas the embryonic cell within itself, they are forming the germinal disc. So cleavage is a stage in the development of embryo, which starts just after the fertilization, or say with the fertilization in the developing embryo, and ends at the MVP, that is mid-blastula transition, a stage where the DNA of a blastomere, it start dividing. It's become active. It's no more a heterochromatin. Cleavage probably is a stage which bring the original cytoplasmic nucleoplasmic ratio to the we bring it originally as it is originally present. And if we compare the embryo development with the evolution is also development of individual from a single cell bacteria to the multicellular organism, the most advanced of them being mammals. So if embryo is a repetition of that process, that is from a single cell, that is zygote, represented by bacteria, to the complete newborn, which is represented by an adult male of the mammals, then cleavage probably falls in the evolutionary ladder at the stage of colonial organism. Those are porifers, the first subphylum in the invertebrates. This is an assignment for you, the students. So this is all about cleavage, preceding to which is fertilization and which is succeeded by blastocyst formation. Thank you very much.